My name is Gao Peng. I'll introduce the paper submitted to iMeta. The title is Photoaffinity Probe-Based Antimalarial Target Identification of Artemisinin in the Intraerythrocytic Developmental Cycle of Plasmodium falciparum. Artemisinin constitutes one of the few antimalarial treatments that exhibits effectiveness throughout the asexual life cycle of the parasite. In this work, we identified the protein targets of ART at the ring, trophozoite, and schizont stages using an ART photoaffinity probe, AP, and conducted extensive mechanistic studies by integrating target validation, phenotypic studies, and untargeted metabolomics analysis. Here are the highlights. 1. We identified the targets of artemisinin at different stages of the intraerythrocytic cycle of Plasmodium falciparum using a photoaffinity probe of artemisinin. 2. Artemisinin can interact with parasite proteins in vivo through both covalent and noncovalent mechanisms. 3. Artemisinin may exert its antimalarial effects by interfering with the parasite's protein synthesis, glycolysis, and oxidative homeostasis pathways. 4. This study provides fresh insights into the mechanisms underlying artemisinin's antimalarial effects and its protein targets. In this work, we identified the interacting protein targets, including both covalent and noncovalent binding targets, of ART at the ring, trophozoite, and schizont stages using an ART photoaffinity probe, AP, based on the ABPP technology. A total of 451 potential target proteins were identified. The trophozoite stage has the largest number of targets. And the results also suggest that in addition to heme activation-mediated covalent binding to proteins, noncovalent binding of ART to parasite proteins also occur. Bioinformatics analysis showed these targets have multiple physiological functions and are involved in diverse biological processes. The GO enrichment analysis revealed that the targets of the ring stage and trophozoite stage are mainly involved in protein catabolic process, proteolysis, and other processes, while the targets of the schizont stage are mainly involved in peptide metabolism, translation, and other processes. In addition, the translation, proteolysis, peptide metabolic and biosynthetic processes were enriched at all three different stages. Falciparum elongation factor 1 alpha was identified as a potential target at all three stages. We expressed the recombinant PFEGF1 alpha protein and verified its interaction with AP. In summary, ATS may bind to PFEGF1 alpha, resulting in the inhibition of protein synthesis and thus antimalarial effects. We then went on to examine the impact of ART on parasite glycolysis. The results suggest that artemisinin may bind to glycolytic enzymes to inhibit their activities, thereby interfering with the glycolytic process of parasites, and thus exerting antimalarial effects. The results suggest that ATS may also bind to important peroxidative proteins involved in the maintenance of redox homeostasis in parasites both covalently and noncovalently, potentially affecting the redox homeostasis of parasites. The untargeted metabolomics analyses suggest that ATS can extensively interfere with multiple metabolic pathways of intraerythrocytic parasites, which may directly or indirectly converge on the collective antimalarial effects of ART. Summary. In this work, we comprehensively identified the protein targets of artemisinin at the ring, trophozoite, and schizont stages of Plasmodium falciparum in situ using an active photoaffinity probe, and investigated the binding mode. Our results suggest that ART may exert its antimalarial effect by blocking parasite protein synthesis, interfering with the glycolytic energy supply pathway, and disrupting redox-related processes. This work provides new insights for the study of the antimalarial mechanism of artemisinin, which is necessary to optimize current antimalarial drug regimens and offer novel solutions to mitigate artemisinin resistance.